Dear students, this is the second session of nucleic acids of 6th semester chemistry DSc 2B, University of Mysore. Contents of this video are structures of nucleotides, structure of DNA and RNA, DNA double helix structure, a ribonucleic acid and types. Structures of deoxyribonucleotides. So these are the structures of deoxyribonucleotides: DAMP, DGAMP, DTMP, DCMP. Deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxythymidine monophosphate, deoxycytidine monophosphate. In DAMP, nitrogenous base is adenine. In this case, base is guanine. In this case, base is thymine. Here it is cytosine. In all the four deoxyribonucleotides, sugar is deoxyribose. Structures of ribonucleotides. Here again, four ribonucleotides are there: AMP, GMP, UMP and CMP. Base is adenine, guanine, uracil, cytosine, and sugar content is ribose. All are ribose. Structure of DNA and RNA. This is the structure of DNA, in which sugar is deoxyribose. Two sugar units are linked together by phosphodiester linkage, and these are nitrogenous bases: adenine, thymine and guanine. This is the structure of RNA. Here the sugar is ribose. Two sugar units are linked together by phosphodiester linkage. These are the nitrogenous bases: cytosine, guanine and uracil. DNA double helix structure. The double helical structure of DNA was proposed by James Watson and Francis Crick in 1953. He got Nobel Prize in 1962 for this work. This is the structure of DNA. DNA is a polymer of deoxyribonucleotides. It is composed of monomeric units, namely deoxyadenylate, deoxyguanylate, deoxycytidylate, deoxythymidylate. DNA is a right-handed double helix. It consists of two polydeoxyribonucleotide chains twisted around each other on a common axis. This is that common axis. The two chains are antiparallel, that is, one chain runs in the 5'3' direction. This is that chain, while the other in 3'5' direction. This is the one. The width of a double helix is 20 angstrom unit or 2 nanometer. This is the width. Each turn of the helix is 34 angstrom or 3.4 nanometer with 10 pairs of nucleotides, each pair placed at the distance of 3.4 angstrom unit or 0.34 nanometers. Each strand of DNA has a hydrophilic deoxyribose phosphate backbone on the outside of the molecule, while the hydrophobic bases are stacked inside towards the axis of the helix. This is the phosphodiester linkage bond, and these are the bases. Each base forms hydrogen bonds with the one directly opposite it, forming base pairs. The AT base pair has two hydrogen bonds, while GC pair has three hydrogen bonds. Hence, the two polynucleotide chains are complementary to each other. The genetic information resides on one of the two strands known as template strand or sense strand. The opposite strand is antisense strand. The double helix has major grooves and minor grooves along the phosphodiester backbone. Major groove and minor groove. Ribonucleic acid. Ribonucleic acid is a type of molecule that consists of a long chain of nucleotide units. Each nucleotide consists of a nitrogenous base, a ribose sugar, and a phosphate. RNA is very similar to DNA but differs in a few important structural details. In the cell, RNA is usually single-stranded while DNA is usually double-stranded. RNA nucleotides contains ribose while DNA contains deoxyribose. And RNA has the base uracil rather than thymine that is present in DNA. Chemical structure of RNA. It has ribose sugar, phosphate group and nitrogen bases adenine, guanine, cytosine, uracil. This is the structure. Types of cellular RNA. There are three types of cellular RNA. One is ribosomal RNA, rRNA. Second one is messenger RNA, mRNA or template RNA. Third one is soluble RNA, sRNA or transfer RNA. Ribosomal RNA. Ribosomal RNA, as the name suggests, it is found in the ribosomes. It comprises about 80% of the total RNA of the cell. It is involved in cellular synthesis of protein. It provides structural framework for ribosomes. Messenger RNA. It carries the information for protein synthesis from the DNA to the sites of protein formation, hence the name messenger RNA. It consists of only 3 to 5% of the total cellular RNA. Each gene transcribes its own mRNA. When one gene codes for a single mRNA strand and the mRNA is said to be monocistronic. Sometimes several adjacent cistrons are transcribed into a single mRNA molecule called polycistronic or polygenic. Transfer RNA. The second most common RNA in the cell is transfer RNA. It is also called soluble RNA. It constitutes about 10 to 20% of the total RNA of the cell. It serves as the carrier of a specific amino acid to the site of translation. There are at least 20 species of tRNAs corresponding to 20 amino acids present in protein structure. MCQs. The first question is, the information for protein synthesis is stored in nucleic acids. Second is, nucleotides are linked together to form nucleic acid through which type of bond? That is phosphodiester bond. Third question is, the repeating units in both DNA and RNA are called nucleotides. Fourth is, the sugar that is in the backbone of DNA is deoxyribose. Fifth question is, what nucleotide base does RNA contain that DNA does not? Uracil. Thank you.